Hi everybody, this video is going to focus on eukaryotic DNA storage, specifically on how DNA is stored within a human cell, because remember human cells are eukaryotic cells. So let's go ahead and draw a cell here. Within this cell we have many different structures, many different organelles such as ribosomes, the mitochondria, we've got endoplasmic reticulum, the smooth and rough, but we've also got a nucleus, and within the nucleus of this cell there's going to be a lot of DNA. Now, DNA is a really long structure. It's a very, very large structure. So how do you fit this long structure into such a small nucleus of the cell? Well, that's where these kind of circles or aggregates called histones. So I'll just write an H. You have all of these histones. Now, this DNA is thus going to go and wrap around these histones to cause kind of this supercoiling, and once we compact once we wrap these histones with DNA, you can tell that we can fit a lot more DNA into this structure instead of just have this one long strand. So what we're going to be doing is we'll take this long strand of DNA and wrap it around these histones to form a more compact, tightly organized structure. Now, these histones are going to be broken down further into 8 building blocks, or 8 kind of Lego units, or however you want to describe it. Now, let me change color here. These 8 units are going to be kind of double. So we have 2 H2A units. So both of these are going to be H2A units. We're going to have 2 H2B units, 2 H3 units, and 2 H4 units. All of these are going to combine into forming one histone unit. So each histone has 2 H2As, each histone has 2 H2Bs, 2 H3s, 2 H4s, etc., etc. Okay, so each of these units can be broken down further. So this unit is going to be made up of proteins. Remember, proteins can be either charged, uncharged, charged or uncharged, and each charge could either be positive or negative depending on what protein is found. So we're going to remember that these histones have a lot of proteins that have a charge, and more specifically, they're going to have a positive charge. So some of our positively charged proteins are going to include our lysine and arginine. So our lysine and arginine proteins are usually the positively charged ones, and if these histones are made up of a lot of lysine and arginines, then that means that these histones are generally going to have a positive charge overall. So let's go ahead and draw this over here. We have one histone unit. It is going to have a lot of positive charges around it, because it's made up of a lot of lysine and arginine that make up all of these units, and all of these units make up a histone. Alright, so then in comes our DNA strand. So let's draw another DNA strand. Let's separate kind of over here. So we have this long DNA strand. I'm going to tell you that DNA is negatively charged as a whole. So negative and positive charges are going to attract each other. Thus, we have an attraction. So when our DNA meets our histone, we're going to have an attraction, and it's going to wrap itself around the histone. This is how eukaryotic DNA storage occurs at the very basic level. One last thing of note is, now that we've created a compact structure of DNA and histones, we're also going to expand it into including a more complete picture. So not only do we have these red histone octamers that is going to compose our stored DNA, we're also going to have another subunit, because remember we had H2A, H2B, H3, H4. Well, what about H1? Well, we're going to have these other subunits called H1 subunits, And these protein kind of structures, these H1 structures, are going to be on the DNA themselves. DNA is not going to wrap around them. These are kind of the beads on a string. So that's where the term beads on a string comes in. We're going to have this string, which is our DNA. We're going to have these beads located on it, which are the H1 subunits. And then eventually we're going to find a histone subunit. Our DNA is going to wrap a couple times around it and cause a nice compact structure. This, let's see, this right here is going to be called a nucleosome. So the DNA, along with a histone complex wrapped around it, is going to be a nucleosome. While our chromatin is going to include our proteins, DNA, and RNA as well. Thank you all for watching. If you have any questions, be sure to ask. Otherwise, make sure you stay tuned for our next video in the biochemistry series.